today we are considering a term that is called Chemerism. Chemerism. I want to look at what it means, the effects, and the control of Chemerism. Chemerism is a rare phenomenon where two or more different cells population, often from different individuals, coexist within the same person. This can occur naturally, such as in the case of twin, or artificially, through medical procedures like organ transplantation or bone marrow transplantation. Chemerism can affect DNA paternal testing in several ways. So we're looking at the different effects of Chemerism on DNA paternal testing. So we have been able to establish that Chemerism is a rare phenomenon where two or more different cells population, often from different individuals, coexist within the same person. So different cells from different individuals will now coexist within the same person. This can occur naturally, such as in the case of twins, or artificially through medical procedures like organ transplants or transplantation or bone marrow transplantation. So this can actually affect paternity tests or paternal tests in several ways. Now let's look at the effect of Chemerism on DNA paternal testing. 1. False negatives. If the Chemeris DNA contains cells from another individual, a DNA paternal test may not detect the biological father DNA, resulting in a false negative. So if the Chemeris DNA contains cells from another individual, a DNA paternal test may not detect the biological father's DNA, resulting in a false negative. Number 2, false positives. Conversely, if the Chemeris DNA contains cells from another individual who is related to the alleged father, a DNA paternal test may incorrectly identify the chimera as the biological father, resulting in a false positive. So there's tendency to have a false negative test and a false positive test if the cell of the paternal has chimera cells. So I said, conversely, if the chimera's DNA contains cells from another individual who is related to the alleged father, a DNA paternal test may incorrectly identify the chimeras as the biological child, resulting in a false positive. Number 3, mixed DNA profiles. Chemerism can result in mixed DNA profiles, making it difficult to interpret the result of a DNA paternal test. So this is a great complication. So when it can lead to mixed DNA profile, the chimeras may result in mixed DNA profile, making it difficult to interpret the result of a DNA paternal test. Number 4, increased risk of misidentification. Chemerism increases the risk of misidentifying the biological father or child, which can have significant emotional, social, or legal consequences. So it can lead to increased risk of misidentification. So where the Chemerism increase the risk of misidentifying the biological father or child, which can have significant emotional, social, and legal consequence. Now, what is the control measure to minimize the effect of Chemerism? Control measure to minimize the effect of Chemerism. Use of multiple DNA samples. Collecting multiple DNA samples from different sources, e.g. the blood, the saliva, the tissue, can help identify and account for Chemerism. So collecting multiple DNA samples from different sources like the blood, the saliva, the tissue can help identify and account for chimerism. Number 2, advanced DNA testing techniques. Using advanced DNA testing techniques such as next generation sequencing can help detect and analyze mixed DNA profile. So using advanced DNA testing techniques such as next generation sequencing can help detect and analyze mixed DNA profile. Number 3, interpretation by experienced professionals. Interpretation by experienced professional. Having experienced professional interpret the result of DNA paternal test can help identify potential tissue issues related to chimerism. So having experienced professionals interpret the result of DNA paternal test can help identify potential issues related to chimerism. Number 4, use of additional identification methods. Using additional identification methods such as fingerprinting or other biometric analysis can help confirm identity and reduce the risk of misidentification. So using additional identification methods such as fingerprinting or other biometric analysis can help confirm identity and reduce the risk of misidentification. Number 5, disclosure of chimerism status. Individuals who have undergone medical procedures that may have resulted in chimerism should disclose this information when undergoing DNA paternal test. Disclosure of chimerism status. Individuals who have undergone medical procedures that may have resulted in chimerism should disclose this information when undergoing DNA paternal testing. Number 6, development of chimerism-specific DNA tests. Researchers are working on developing DNA tests that can specifically detect and analyze chimerism. So we have looked at this important phenomenon, chimerism, which can actually alter or give a false DNA paternal test results. It has been discovered that there are series of paternal tests that are not really true because of this phenomenon, chimerism. We say it's a rare phenomenon where two or more different cells population, often from different individuals, coexist within the same person. So this can occur naturally, such as in the case of twins, or artificially through medical procedures such as organ transplantation or bone marrow transplantation. So it can affect DNA paternal test by giving us false negatives or false positive, and also can lead to mixed DNA profiles and increased risk of misidentification. Also, we said that under the false negatives, that if the chimera's DNA contains cells from another individual, a DNA paternal test may not detect the biological father DNA, resulting in a false negative. And also that it can give false positive result. If the chimera's DNA contains cells from another individual who is related to the alleged father, a DNA paternal test may incorrectly identify the chimeras as a biological child, resulting in a false positive. So there are tendency to have wrong DNA results that may not be correct because of the effect of this chimera. And we have talked about the control measure to minimize the effect of chimera. So using of multiple DNA samples, advanced DNA testing techniques, that this by applying advanced DNA testing techniques, the interpretation by experienced professionals, and the use of additional identification methods. And we said that the disclosure of chimerism status, the individual who have undergone medical procedures that may have resulted in chimerism should disclose this information when undergoing DNA paternal testing. And lastly, we talk about the fact that development of chimerism-specific DNA testing, the researchers are working earnestly on developing DNA tests that can specifically detect and analyze chimerism. So in conclusion, chimerism can significantly impact the accuracy of DNA paternal testing. However, by understanding the effect of chimerism and implementing control measures, we can minimize the risk associated with this phenomenon and ensure the accuracy and reliability of DNA paternal testing. So with this, we have come to the end of this study. Thank you.